Good afternoon. My name is Alex Huang. I'm a professor of pediatrics at Case Western Reserve University School of Medicine. It's my pleasure to discuss some of our ongoing research trying to enhance sarcoma immunotherapy. Here's the disclosure information. I will briefly introduce an investigator-initiated trial targeting sarcoma tumor microenvironment using integrin therapy. And in the remaining time, I will discuss our molecular determinants of cryoablation efficacy in sarcoma. Historically, we have focused primarily on identifying tumor intrinsic vulnerabilities, including genetic alterations, cell cycle progression, protein synthesis, metabolomic, and nutrient responses and stress response as a way to target cancer. And this has led to the combination chemo, surgery, radiation therapy for many of the cancers. However, recently, we have now increasingly understand that non-cancerous cells in the tumor microenvironment also contribute and potentially are vulnerabilities we can target. And today I will focus on immune suppression, invasion, and resistance. Tumor microenvironment consists not only of dividing cancer cells, but there can also consist of multiple host-derived cells, including stroma, vasculature, as well as hematopoietic lineage cells, including macrophages, or dendritic cells, or myeloid suppressor cells. These myeloid lineage cells are thought to be predominant infiltrates in many of the pediatric cancers as opposed to adult cancer. And the presence of these myeloid lineage cells, including CD68 positive macrophages or CD1A dendritic cells, pertain to poor survival in some sarcomas, such as osteosarcoma. So today I will focus first on describing integrin therapy for metastatic osteosarcoma. And this is the discovery of the surface expression of VCAM1 or CD106 on metastatic osteosarcoma and their interaction with alpha-4 beta-1 integrin or VLA4 on tumor-associated macrophages. VCAM1 or CD106 has been described to be important in metastatic breast cancer using 4T1 cell line, for example, showing that the 4T1 cells that evade the lungs or the bones overexpress on the surface VCAM1, and that transmit a pro-survival signal into the tumor cells to allow them to thrive in a metastatic niche. We interrogated publicly available databases to show that in some osteosarcoma cells, VCAM1 is overexpressed. Using syngeneic mouse model, in which K7M2 is a derivative of K7 derived from a Bell-C mouse, and K7M2 has a strong propensity for pulmonary metastasis when injected intratubularly. We found that K7M2 on the surface, as well as messenger RNA, overexpress CD106, and that is correlated with their ability to metastasize to the lungs using bioluminescent imaging, as well as a tumor-specific mRNA marker, GP70. When we knock down VCAM1 or CD106 on K7M2, we can show that that has strongly reduced the K7M2's ability to metastasize to the lungs, indicating the importance of VCAM1 expression on the surface. Next, we asked the question whether that VCAM1 interaction is via macrophages in the tumor microenvironment. We did this by performing intratubular injections of K7M2, followed by amputation at the time of metastasis to the lungs, and deplete macrophages in the lungs, either intranasally or IV, showing that macrophage depletion was able to rescue some of the mice from developing metastatic cancer. We then asked the question whether in the presence of macrophages, whose function is also important in anti-infectious activity, using antibody that blocks this interface between alpha-4 beta-1 and VCAM, as well as a mimicking peptide that interacts in the surface between alpha-4 and VCAM1, can lead to the same outcome. And indeed, we found that in the presence of metastatic cancer or osteosarcoma, we can use anti-alpha-4 antibody or this anti-VCAM peptide, leading to 80 to 60% of mice disease-free. In this lung microenvironment, what we found is that in a VCAM high environment, there's an infiltration of tumor-associated macrophages involving CD206 expression. In the VCAM low tumor microenvironment that actively rejects the osteosarcoma, NK cell accumulates as well as CD206 low myeloid cells. These discoveries led to the development of a natalizumab phase 1, phase 2 clinical trial in which natalizumab is already FDA approved for multiple sclerosis and inflammatory bowel disease targeting the alpha-4 integrin of migrating lymphocytes. And we designed this phase 1/2 clinical trial targeting unresectable pulmonary metastatic osteosarcoma between the age of 5 and 30. And this is with the support of Biogen. The IND has been approved, and we are now waiting for final approval for IRB, and is slated to open later on in Q3 of this year. Let me describe another new area of research that involves a discovery of molecular determinants of response to cryoablation in sarcoma. Cryoablation is a FDA-approved procedure whereby a probe is inserted into a tumor site or sometimes a nerve bundle and pump ultra-cold liquid nitrogen or argon gas to freeze the tissue. And the water crystal developed in the frozen tissue disrupts the membrane integrity, leading to elaborations of molecular pattern molecules that may serve as an immune adjuvant, in addition to destroying the tissue of interest. In fact, cryoablation has been used in combination with immunotherapeutics, and ongoing evidence suggests that as opposed to apoptotic cell death, cryoablation induces necrotic cell death, whose debris from the tumor are able to activate the immune system, leading to the development of an abscopal effect in which you're seeing immune responses against non-cryoablated tumors away from the primary cryoablated tumor mass. However, the challenge remains that the clinical response observed using cryoablation is inconsistent, and there are also paradoxical outcomes with partial versus complete tumor ablation, in that partial ablation sometimes result in better outcome than complete ablation. So we wanted to know the molecular basis of this. And in thinking about this, sting and C-gas pathway has been implicated as one of the primary ways by which cryoablation works. The sting C-gas pathway is an innate sensing pathway where double-stranded DNA activates the C-gas in producing dinucleotide CGAM, which is a ligand to cause oligomerization of sting, that in turn activate TBK1 and phosphorylase RF3, which translocates and activates a downstream transcriptomic machinery, including the production of type 1 interferon that drives the immune response. As a result, multiple clinical trials have used sting agonists trying to activate the myeloid-derived sting pathway in activating these type 1 interferon and subsequent immune responses. We asked the question whether a cryoablation-induced RMS tumor can activate sting in similar fashion. We use a mouse model of rhabdomyosarcoma 76-9, where Mohammed took the cell palette and going through cryoablation procedure and feed the tumor lysate into bone marrow-derived dendritic cells or macrophages. In all cases, the tumor lysate from cryoablation were able to induce the production of immune reactive genes, including class 2, CD80, 86, and IL-12 beta, both in macrophages as well as in dendritic cells. And that production of interferon beta and downstream sting pathway is dependent on the myeloid cells sting, as sting knockout derived macrophages or dendritic cells were unable to do so. We then took it in vivo and showed that if you inoculate 76-9 into B6 mice, and after day 10, when the tumors are quite sizable in this aggressive tumor model, and we applied 1 round of 3 repeated cycles of cryoablation on day 11, this led to a very robust reduction in tumor growth. And that also subsequently induces the upregulation of class 2, CD80, 86, and IL-12 in the dendritic cells found within the tumor microenvironment. Concomitantly, there's a decrease in the overall lung metastatic lesions of these animals undergoing cryoablation. We subsequently described that these reduction in tumor is not due to solely the destruction of cryoablation on tumor kinetics, because similar observations cannot be recapitulated in nude mice. And these tumor, although the tumor initially undergo regression and it very quickly caught up. Similarly, in B6 mouse where the tumors respond, if we were to deplete CD4 or CD8 T cells, this led to the relapse of tumors very rapidly. Similarly, in mouse undergoing cryoablation, we also see enhancement of T cell accumulation in the cryoablated tumor bed, as well as their function. We asked the question whether, besides the myeloid cells derived from hematopoietic or elements of the host, tumor cells also produce a STING. Mohammed was able to show that these 76-9, when they're fed in vitro with 76-9 cultures, they're also too able to produce interferon beta, and that when you knock out the genes, the STING gene in the 76-9, they're unable to. And this is just to show you some of the downstream signaling pathways of phospho-RF3 present in wild type RMS, but is absent when we are using CRISPR to knock out the STING. So it led us to then ask the question, what is the relative contribution of STING pathway activation in myeloid cells versus STING activation pathway in the tumor cells themselves subsequent downstream to the cryoablation? And here is the surprising data. If you were to use wild type tumor, which has intact STING, and inoculate into a STING knockout animal, we see a deficiency of tumor protection. Although that is not completely abrogated, you still have partial protection against the tumor growth. However, if you were to use a wild type mice, which have all the STING in the myeloid component, but now the tumor itself is STING knocked out, you lost that ability of the cryoablation to provide some protection of the animals against tumor growth completely. Whereas in the STING knockout mice, where none of the myeloid component or vascular component have STING, the presence of STING in the wild type tumor, in the tumor microenvironment at the time of cryoablation, are able to somewhat induce a reduction in tumor, whereas the other groups were not able to. This is correlated with the ability of cryoablation to diminish metastatic lesions in the lungs, so that knockout of STING in the tumor itself resulted in multiple lung nodules, whereas the wild type STING present the tumor cells, irrespective of whether there is STING in the host immune response, were able to reduce the pulmonary metastasis after cryoablation. And surprisingly, same thing too, in the high dimensional t-SNE analysis, it shows that following cryoablation, both lymphoid and myeloid environment demonstrate very similar immune landscape, whether or not the host has STING, but it's solely determined on whether the STING is present or absent on the tumor cells. And this goes to show you some confirmation of T cell subsets in terms of interferon gamma production. We're now beginning to look at multiple tumor sites and to look at the signaling pathway of STING in the tumor, and to see whether or not there are specific reasons why certain tumors contain or not contain STING, And just to show you example, for example, K7M2, as mentioned earlier, does not itself express STING. And our prediction is that in this tumor model, cryoablation will not be useful, whereas replenishment of the STING pathway in this mouse model of osteosarcoma will restore its therapeutic efficacy following cryoablation. So in summary, we show that cryoablation suppresses primary metastatic rhabdomyosarcoma in a T-cell dependent manner, that cryoablation can induce myeloid cell differentiation and interferon beta production in the myeloid cells, that RMS 769 contains functional STING, and this functional STING is important in changing the immune landscape, and that the responsiveness of the animals to cryoablation tracks with tumor STING status rather than the host myeloid STING status. And we are now beginning to look at the possibility whether looking at STING as a biomarker and its related genes as a biomarker therapy response in cryoablation in multiple tumor types. Just want to thank the people in the laboratory for carrying out these experiments, and we thank for funding support. Thank you very much, and happy to take any questions during the panel discussion.